Hello everyone, my name is Tel Kreutzbergers and I'm going to introduce this, give the first talk of this session on decoding and representational dynamics in MEG and EEG. Now, the general term MVPA and decoding are somewhat used interchangeably. So MVPA stands for multivariate pattern analysis. You also see multivoxel pattern analysis sometimes. And decoding is one of the tools in the MVPA toolbox that refers to predicting the condition from the data. And MVPA and decoding methods have been used for the past 20 odd years in fMRI, and they're well established in that field. But they're still relatively new in MEG and EEG, and this is going to be the focus of my talk today. In MEG and EEG, on the contrary, the univariate methods are very well established and have been used since the 1970s. And because these methods have been very well established, this can lead to confusion, and I'm going to go into a couple of these sources today. MVPA allows you to study representational dynamics. It allows you to ask different kinds of questions. And because of this, and because of the sensitivity of the approach, it is rapidly increasing in popularity. And we're seeing more and more papers come out using MVPA. And recently, we've seen more toolboxes on MVPA specifically applied to MEG and EEG. Now in this talk, I'm gonna go over these points. I'm gonna give you a quick introduction why you want to use classification methods. So why would you want to use decoding rather than univariate methods? I'll go into an example pre-processing and decoding pipeline specifically tailored to MEG and EEG. And then I'll go into representational dynamics and a representational similarity analysis method. I'll give some notes on some open questions, some perspectives, and some things to watch out for when you're decoding. And that wraps it up for today. So first of all, why would you want to use classification? Well, the main reason is that you can use this to answer different questions. So, and the different questions come out of the different way of looking at the data. And the main difference is the univariate versus multivariate. And I'm going to go into these examples a little bit in the next slides. MVPA allows you to do a non-directional approach, and it allows you to look for information rather than for activation differences. The method is highly sensitive and powerful, so you can get results more easily. And finally, it's spatially unbiased and data-driven, and I'll go into detail about what that means as well. So the first one is univariate versus multivariate. This is the most straightforward one. So with MVPA, we are looking at multiple variables and not at a single variable at a time. So look at these examples on the screen. Here we have two subjects. Red and green dots represent individual trials, for example, in an experiment. And on the two axes, we have the activation on each channel. So here, two channels, for example. Now, in a univariate scenario, you would look at the difference between the mean of red and the mean of blue on each channel separately. And you may conclude that there's no difference because these distributions overlap quite a bit. Now, in a multivariate approach, you would look at how well they are separated by taking into the information at all channels combined. So here we can see that these two distributions can actually very nicely be separated if you integrate the information across the two channels. The second example of MVPA compared to univariate analysis is that you generally look for non-directional effects. Well, in a univariate analysis, you would look at the difference across subjects. So you would average all your activation across subjects. And in this example, because the subjects are polar opposites from each other, you would conclude that there's no difference because the red and the blue on average across subjects have the same activation on channel 1. In MVPA, you would ask the non-directional question, which is red different from blue? And this is the case here for both subjects. So for both subjects, red and blue are easily separable, and you then integrate the information at the group level. So here you you answer this question by saying, yes, they are different at the group level, because in each subject we can separate the conditions. And finally, we look for information rather than activation. So what I mean by that is the following. Here we, in an app, we have examples of 4 conditions. So we have red, blue, green, and yellow. And in an activation-based approach, you would look at which conditions are associated with stronger activations on a particular channel. So here on channel 1, we may conclude that we have an increased response for blue, yellow, and green conditions. So that would mean that the response of these channels reflects blue, yellow, and green. But from an information-based perspective, we can also conclude that A is different from B, C, and D. So it might actually be more informative to say that this response encodes something about A rather than B, C, and D. So I hope that you see this difference in the type of question that we're asking here. Now, finally, another reason to do this MVPA is because the analysis is often spatially unbiased and data-driven. So, for example, in EEG, we generally, in a univariate approach, would select channels or ROIs to look for activation. Now, here's a study by Céline Petit and others where we had 20 subjects, and we here illustrated the activation at the scalp for each of these subjects. So you can see that they are really, really different across each subject. So when you aggregate this information all together, you might conclude that there's no reliable pattern across subjects. But if you use MVPA in this case, we can look at individual differences in subjects here and then aggregate the information at the group level. And indeed, what Petit found was that we could decode these individual subjects with high reliability. So for most of these subjects, we had accuracy above chance, even though there was no consistent pattern across subjects that underlie these differences. So these are the reasons that you might want to use a classification approach rather than an activation-based univariate approach. Now I'm going to go over a very simple decoding pipeline. So this is the basics of what you would do in a classification approach. And I'm going to use MEG and EEG, so time-varying neuroimaging data, as an example. So this is what I do in my research most often. And generally, a standard pre-processing pipeline applies. So you can use all the methods that you're familiar with in MEG or EEG pre-processing. Most of them would work similarly, would be similarly helpful for an MVPA approach. One exception may be that I often don't do a channel selection or exclusion, because a classifier can actually use the information and can exclude channels itself. If there's no information in a channel, it might just assign a 0 weight. But also, if you include a noisy channel rather than remove it, it can actually help the decoding. So what I mean by this is, consider the following example. Here we have 2 signals that are that have co-varying signal in 2 channels. And one channel, channel 1, is encoding the signal of interest, so the slight difference between the conditions. But channel 1 is also measuring some noise, and channel 2 in this case is measuring the exact same noise. So in this case, it's very straightforward to see that you can reconstruct the signal by subtracting the 2 channels. So one channel can give you, one noisy channel can actually help the classification. Another common pre-processing step is excluding trials, and I often avoid this in my pre-processing pipelines because it can result in unbalanced data, so that you don't have enough, don't have the same number of trials for each class in your decoding, and this can lead to biased classifiers and a whole other range of possible problems down the track. So I generally advise against doing this. But if you must exclude trials, then you have to make sure that you have balanced data to go into the classifier. So this is an acceptable default pre-processing pipeline, but most pre-processing pipelines work fine, and there's no optimal pipeline in the literature that exists at the moment. So there's no real standard default pipeline that you should use. Now, after you pre-process your data and you epoch your data into trials, we train a classifier. And here I illustrated the activation space in channel space. So here is a three-dimensional picture where each dot reflects a single trial in our experiment. So we have red and blue trials in our experiment as an example. And each point here is the activation on 3 channels. So you can see that this results in these three-dimensional clouds of points. And for EEG, for example, this would be a 64-dimensional space if you use 64 electrodes. For MEG, it's often much higher, so you would get 160 or 300 dimensional space. But we can only reasonably visualize 3 dimensions, but you have to imagine scaling this up to many more dimensions. Now we're going to train a classifier to find a distinguishing hyperplane that separates these conditions optimally. And several classifiers have been used in the literature. Here's 3 really popular ones: Gaussian naive Bayes, linear discriminant analyses, and support vector machines. And these classifiers are all linear classifiers. What they have in common is they all will try to find the optimal separating hyperplane. They differ a little bit in the assumptions and how well they do with high dimensional data. So when you have a lot of dimensions and maybe a few trials, so these classifiers differ there. Would refer you to the literature to see what the exact different assumptions are here. But they generally result in a very similar hyperplane that separates the 2 conditions. So here we have a 3-dimensional hyperplane that separates red and blue dots as best as it can. This is called, this is the training phase of the classifier. So here we train the classifier to find this separating hyperplane. Then we take the trained classifier, so we take this hyperplane and we test how well it predicts unseen data. So we have some data that we did not use for training, and we project this onto this hyperplane, like so. And then we count how often the points fall on the right side of the hyperplane. So in this case, we see that most blue points fall on the right side of the hyperplane, most red fall on the left. There's maybe some errors in there. And this gets us a prediction accuracy on the unseen, on the held out data. So what we did here was we kept out some part of the data, here maybe 25% of the data we used for testing the classifier, and the 75% of the data we used for training the classifier. And the resulting accuracy on the test predictions may be something like 73%. Then we repeat this process, holding out a different set of data each time. So we repeat this here 4 times, and each time we test a different part of our data, and we retrain the classifier. We compute its prediction accuracy on the held out data. So here we have 4 different prediction accuracies: 73, 61, 57, 69%. And we average all these accuracy together, and this gives us our mean cross-validated decoding accuracy. So cross-validation refers to splitting the data up in training and testing sets repeatedly, and training the classifier on part of the data, testing the classifier on the held out data. And this last number, this final number, is our result. So this is our cross-validated decoding accuracy. Then for neuroimaging data, we repeat this whole process for example, for different brain regions in fMRI, or for different time points in MEG. So you can repeat this at different points in time, or using different ROIs in fMRI, for example. So in the MEG and EEG example, this is what is called sliding window decoding. This means we slide a window over the response to individual trials, and at each time point in the response, we repeat this cross-validation approach. So here we have a couple of example EEG trials where we have a number of channels and we have time points in response to the stimulus. And at time t, we take all the activation of all the channels and we vectorize them. So now we have patterns of activation. Here, the color of each box refers to an activation in a channel. We assign the trials to training and testing data, and we train a classifier, we test it, we repeat a couple of times to have cross-validated data, and then we store the accuracy, the mean cross-validated decoding accuracy at time point t. And then we slide this window to the next time point. And we repeat this process over and over again so that we get a performance at each time point in the response. And that often looks very similar to something like this. So here we have chance accuracy, what you would get if the classifier was purely guessing. We have time point 0. This is when we present the stimulus. So when, for example, a picture comes on the screen. And on the y-axis, we have classification accuracy. And what we see, what we hope to see, is that classification accuracy is at chance before 0. This is our sanity check. It should be a chance. There's no information in the brain signal at this point because there's no image on the screen yet, for example. So we expect our classifier to be a chance. Even sometime after the image came on, we still expect it to be a chance as the information starts coming in. And then at some point, we hope that our classification accuracy goes up. And that's what we illustrate here. So here we have a peak around here, and then we often see a slow drop off back to chance. So this is the classification performance over time. Now, this is the analysis that you run on one specific subject. Then you repeat this whole process over subjects. So you create decoding over time for every subject. And now you can aggregate these at the group level, and you can test whether your group mean accuracy is bigger than chance at every time point. And this has been done in very different ways in the literature. A popular one is to do a standard t-test against chance level. You can also do a non-parametric sign rank test, for example, or you can do a permutation test where you shuffle the labels and then train your classifier again, and then you get the classification accuracy under the null hypothesis that you can compare against. Now, importantly, because we're testing the classifier, we're testing these accuracies at each time point in the response, we have to correct for multiple comparisons. So in our case, across time, because we have multiple time points. And there's also multiple ways to do this. You can do a very conservative Bonferroni correction. Also very popular is to use the false discovery rate or cluster-based methods, similar to fMRI. And I will refer you to the tutorials and reviews to see how to implement these. So this is a very standard MEG decoding pipeline, and I hope that this sort of illustrates how this process works. Now, in the next part of the talk, I want to take it one step further, and we go into representational similarity analysis. This is a technique that was popularized by Nicolaas Kriegeskort in 2008. And this is a technique where you can test models of information processing. You do this by comparing similarities in brain responses with similarities that are predicted by different kinds of models. So to give you an illustration of this approach, this is a very popular approach in cognitive neuroscience at the moment. We go back to our EEG data again. So we have our response to two different stimuli. Here we have stimulus I and stimulus J. And at each time point, again, we do a sliding window approach here as well. So at each point in time, we take the activation at all our channels for stimulus I and stimulus J. And then we compute some measure of dissimilarity. Now, there's multiple different measures that you can use. So one of the measures is straight up decoding accuracy. So we can use a classification accuracy between two stimuli as a measure of dissimilarity. So the more dissimilar the patterns are, the better the classifier would distinguish between them. We can also use a correlation distance or a Euclidean distance, a cosine distance. All these measures have been used in the literature, and there's papers comparing the pros and cons of all these measures. So we compute some kind of dissimilarity measure between the response to I and J, and we store this in a matrix. So here we have stimulus on the x-axis, stimulus on the y-axis, and each point in this matrix is going to be the similarity between two stimuli. So here we have stimulus I and stimulus J, and the dissimilarity between those goes in this point. And we repeat this for all the stimuli that we have. So we might have here 100 so stimuli, and for all these 100 stimuli, we compare the differences. So this is a very popular approach in visual cognitive neuroscience, because we can, for example, use different images as different stimuli. Now, the color of each of these pixels represents the extent to which these two stimuli are similar or dissimilar. And we repeat this process at every time point again in the epoch. So we get one of these matrices for every time point in the epoch. And we also repeat this across subjects. So for each subject, we get a time varying dissimilarity matrix. So these matrices are called dissimilarity matrices. And then if we want to try to model the information in these matrices, we compare a candidate model to these matrices. So this might be a candidate model, this might be a computational model or a behavioral model that predicts the similarities between two stimuli. So here the color in this model is predicting, so the yellow ones are predicting that there's a very dissimilar response, and the blue squares predict that between those stimuli there's going to be a very similar response. And we compared the model to these time-varying dissimilarity matrices at each point in time. So generally what we do is we take the lower triangle, because these matrices are symmetric. We take the lower triangle of these models, the lower triangle of these models, and we vectorize them, and then we again compute some kind of measure of distance. So often this is done using correlations. So we correlate our candidate model with our time-varying dissimilarity models, like so. So for each subject, we now have a time-varying correlation between the candidate model and the subject's time-varying dissimilarity matrices. And we can analyze these correlations again at the group level, where we test whether the mean correlation is higher, significantly higher than 0. So this is a quick overview of this RSA approach. And I'll give you some examples of what you can do with this. So this is a particularly powerful approach that allows you to compare, for example, different models to neuroimaging data. So here's an example from a study of ours, Susan Wardle in 2016, where we had MEG data. So here we have a neural RDM. So the RDM constructed from MEG responses to different stimuli. And we compared perceptual models and computational models. So this is a behavioral model, and we compared computational models as well. And then for each of these models, we get a time-varying correlation between the neural data and our model's predictions. And here's the results. So we can see that we get some correlations from our retinal envelope model, we get correlations from our perceptual model, and these correlations were significant across time, and some models outperformed other models. So for the details, I'll refer you to these papers, but just to give you an illustration of what this approach can achieve here. You can also directly compare multiple models. So here's a paper that compared a combination of models by using linear regression using these models. And this way you get a beta estimate for each of your models. And this refers to the unique contribution of each of these models to the neural dissimilarity. So this is how you can include multiple models. Here's another study where we had MEG recordings to images, and we compared the neural response to ratings of emotional, on different emotional dimensions. So here we computed behavioral distance matrices based on subjects' responses to individual images, and then we compared them to the neural MEG data. And this gave us, for each of these emotions, a time-varying correlation of when this emotion was represented in the neural signal. You can also use RSA to compare different neuroimaging modalities. So this is what Nico Kriegerskor did in 2008, where he compared the fMRI responses to monkey neurophysiology responses. But it also has been used by, it's been pioneered by Rsa and others in 2014 to compare MEG to fMRI data in humans. And this gave a spatial temporal picture of object representations in the brain by looking at where fMRI and MEG data overlapped and at what points in time in the MEG and at what points in space in fMRI they activated together. Now you'll probably see some more RSA work today, and hopefully this gives you enough of a basic understanding of the method to follow the next couple of talks, and the same with the decoding. So in the last part of my talk, I want to go into some perspectives on decoding methods, and it's these 3 points in particular. So I'll jump into the first point straight away. So the first point is that there's very many possible ways to analyze your data. So because MVPA, and especially MVPA on MEG and EEG, is relatively novel, there's no set guidelines and there's no real consensus on what the optimal approach is to analyze the data. What are the optimal statistics? What are the best ways to train your classifiers, to split up the data? There's no set optimal way. So it often comes down to trial and error, and this can often lead to people trying out many different things. So you have to be really careful here that you do not cherry pick your data and choose you choose the classifier that works, for example. So so hopefully in the future we get a little bit more consensus on what is a good default method on this. And some examples of this are, for example, that there's many different labs that have different traditional pre-processing pipelines, and they often borrow the same pipelines for their MVPA approaches. But maybe there's actually an optimal MVPA approach, or maybe some of these traditional pre-processing pipelines actually do not work as well for MVPA. And there's a couple of examples in the literature of why they might not work. The same for the statistical inference and how to correct for multiple comparisons. There's many different methods that have been used, and there's no real optimal way of doing this. There's no consensus, at least, on what the best way is to do this. And there's a couple of possible misconceptions here as well. So I'm going to give you one example that is related to statistics, and that is how to interpret the onset of decoding accuracies. So this is an example that we included in our 2017 tutorial paper on MEG decoding. Here's 2 traces of object animacy decoding. So we have the blue line, which were stimuli that were presented in a clear condition, and we have a yellow dash line that were stimuli presented in a degraded condition, so we blurred the image. And this was decoding accuracy of object animacy, so whether it was an animal or an inanimate object. So on the y-axis, we have decoding accuracy, and on the x-axis, we have time in the MEG response. The gray bar shows when the image came on the screen. And we saw that around 100 milliseconds there, we saw that the blue line, so clear, presented stimuli, reached significant decoding accuracies, and the yellow line reached significant decoding accuracy a little bit later, just before 200 milliseconds. So you might conclude from this plot that that clear presented items activate earlier or have decodable information available earlier than degraded items. But the difference in decoding onsets does not necessarily reflect a difference in the underlying process. So what I mean by this is there's another thing in this plot that is different between the lines, and that's over there. So the clear objects also had a stronger peak decoding accuracy. And this can be an issue here because the stronger peak can actually lead to, so the stronger signal in general can actually lead to an earlier detectable signal. So here you could confuse the onset with the actual strength of the effect. So we simulated this in a Jacob Neural paper where we have here 3 signals in the plot in the top row. So we have red, blue, and yellow. So blue is a strong signal, yellow is a weak signal. This is simulated data, simulated to have an onset at 50 milliseconds exactly. And then we added Gaussian noise, and we just did a standard t-test on these data. And what we saw was indeed that the strong signal had the earliest onset of significance, even though they were all simulated to have the exact same onset. So what this illustrates, I think, really nicely is that the signal strength will affect your decoding onset. So if you have a difference in the overall signal strength, then you cannot interpret a difference in onset. And one of the ways we tried to get around this in a recent paper, and this was last year in Neuroimage, is using Bayes factors because they actually give you evidence for the null. So here we were one of the first to use Bayes factors in assessing decoding accuracies. And what is nice here is that here we have a dot plotted in each row, referring to a level of a Bayes factor. And what we saw is that with using the Bayes factors, you actually see a very sharp jump from evidence for no decoding accuracy, so evidence for chance level decoding, versus evidence for above chance level decoding. And that was, so maybe Bayes factors can actually help out here because it becomes more clear at what point you actually have no signal and true signal rather than insufficient evidence. The second example of future perspectives that I wanted to point out is temporal variance in the epochs. So this is very specific to MEG and EEG, and this is something that I don't think has been solved yet. So the question is how to deal with temporal variance of the responses. And what I mean by this is that between subjects and between trials, the later processing stages, so later in the epoch, later in the evoked responses, they will have more temporal variance just because there's variance in the brain in general, there's variance in each trial, and there's variance in different subjects and their anatomy, but also their specific individual processing. So to illustrate this, here's, for example, data from our 2017 paper where we were decoding the image. So this is the individual image that we presented. So this is generally a very strong time-locked decoding effect because you can decode this from early occipital responses around 100 milliseconds. So on the top plot here, we have all our subjects. On the bottom plot, I separated them out a little bit. And what you see is that here we have a very strong aligned time-locked peak around 100 milliseconds, and at each subject this peak is present. So every subject has this peak, decoding peak, at the same time. Now in comparison, when we decode a more high-level concept such as animacy, whether the object was animate or inanimate, we have decoding peaks much later in the epoch. So this is around 200 to 400 milliseconds. And what you see is that the data are much more dispersed, and each subject has a slightly different response. So some subjects have a peak around 200. For some subjects, there's a more sustained but lower window of decoding between 200 and 400 milliseconds. So the problem here is that this longer processing or this increased variance later in the epoch can actually lead to a lower average decoding accuracy. So when you want to compare, for example, exemplar versus animacy decoding in this case, you can't, it's not a fair comparison because one is much more nicely time locked to the stimulus onset than the other, which is much more a variable process. So to what extent is decoding accuracy a function of variance in the signal compared to actual effect strength? So there have been some suggestions to dealing with this, and I want to just point out one of them, which is a paper, I think it's published at the moment now, by Diego Vidal, and they introduced a method called temporally unconstrained decoding, where they train multiple classification models that try to integrate information across time. So they, instead of training a model at each time point, they train a predefined set of models that try to optimize the time window per state that is happening. So in our exemplar and animacy case, you might want to train two models, one for exemplar, one for animacy, that try to optimize the time window that they incorporate. But for more information, I'll refer you to his paper. Now, the third point I wanted to jump in is localization of the signal. So this is why you would normally use fMRI if you're interested in the spatial origin of the signal. But if you have MEG or EEG data, there's still some things that you can do. So to find out where in the brain the signal is coming from, because rather than doing the spatially unbiased whole head approach, you can also zoom in to create a map of likely locations and spatial origins of the signal. So one way to do this is to go into source space, and there's algorithms, especially for MEG, this is a popular method, that you can compute a map of the likely sources of channel level activations. So you create a 3D map of possible origins and the activation associated with each origin. And you can actually then feed this map also into classification algorithms. So instead of training and testing your classifier on the whole brain response, you can do it on the specific ROI in the source space, or you can even move this ROI around in source space. So there's some possible issues with this, and the first one is it depends on this. Really, the quality of this will depend on how good your reconstruction of the source space is, and the source space reconstruction algorithms are approximations because this is it's an inverse problem. There's many more possible solutions than we look at. So it really depends on how good the reconstruction algorithm is that you use and how much trust you have in the reconstructed source space, to what extent you can interpret the resulting decoding results. And a separate problem is that whenever you reconstruct source space signal, there's always the possibility, and this is almost guaranteed, that you'll have signal leakage. So this means that if you reconstruct activity in one area, some of the signal will also leak into neighboring areas, and sometimes areas all the way across the brain. And this can be a serious problem if you use classification, because classifiers are very powerful and they will use any information that is available. So there's two papers that are listed here, actually showed that you get physically impossible results due to leakage if you use decoding in source space. So this could be a very potential disastrous problem. Another option is to perform your decoding in channel space and then project classifier weights into source space. And whenever you look at classification weights, you first have to transform them into activation patterns. There's a paper on there showing you how to do that, because the weights are not very easily interpretable. And there's maybe some issue with whether this is even a valid approach, because the forward model describes how magnetic fields are generated or how evoked potentials are generated by a specific source. and not how classifier weights are a result of this. So there could be different physics underlying the same process, especially if you then factor in another transformation that you are doing on the classification weights. So you have to really know then the algorithmic derivation of the weights based on the incoming signal. So there's some problems with decoding in source space, I think, that are very important to keep in mind. Another example that I was showing earlier was that you can use the Rsa approach, and this is called fusion. And this allows you to compare, if you have fMRI and MEG data for your experiments, then you can look at information in space and time by combining the information. And this is using the Rsa approach that I was going into before. However, this is very promising, but it's not applicable to all possible designs. So for RSA, generally you need a condition-rich design. So you can't do RSA if you have 4 or 5 conditions, because then you have too small a matrix to compare across your different modalities. And it's also limited by the strength of the signal and the effect size in both modalities. If you have no result in fMRI, then there's also nothing to relate to MEG, for example. Right, so that brings us to the end of my talk. So in conclusion, the MVPA on MEG and EEG is a very powerful tool to study brain dynamics. It's important to keep in mind that there's different assumptions and there's different interpretations. There's a paper by Martin Hebert and Chris Baker from 2018 that's well worth a read, that goes into these issues in a lot of detail. So most importantly, there's the activation versus information that I started with, and the other one is prediction versus inference, and that is also described in the Hebert and Baker paper. And as I mentioned before, it's relatively new. There's no consensus on the best approach. There's no standard approach that you can rely on. There's increasingly more and more reviews, tutorials, and toolboxes. I've tried to get all of them in a couple of slides, but I'm certain I missed quite a few. And so here's some reviews and meta-analyses. So MEG and EEG specific ones are King and DeHanes, and there's a continuing review on MEG for object representation. There's a number of tutorials out there, mostly for fMRI, but also point you to my tutorial in Journal of Cognitive Neuroscience that describes the MEG approach. And here's an overview of possible toolboxes that you can use to start with your decoding if you're interested in this approach. And with that, I'd like to thank you for your attention.